Genes by Ashley and Jake.
Here we have a green protein, a red DNA molecule, and a purple RNA molecule. And with these molecules, we will be showing the Avery-McLeod and McCarty experiment. In this experiment, Oswald Avery and his colleagues, Malcolm McCarty and Colin McLeod, tested the three molecules shown here that could have been the reason for the transformation of the living non-pathogenic bacteria into pathogenic bacteria, in an experiment previously conducted by Frederick Griffith. These substances were pulled from a heat-killed pathogenic bacteria by Avery, and three separate samples of bacteria were created, each containing an active form of one of the molecules, as shown here. And each sample was tested for its ability to transform live non-pathogenic bacteria into pathogenic bacteria. The only sample that worked, as can be seen here, was the one that contained the active DNA, supporting the statement that the transforming agent was DNA. Here we have a red DNA molecule that is about to undergo semi-conservative replication. In semi-conservative replication, the two parental strands unwind from each other and serve as a template for the synthesis of new complementary strand as shown in blue. This ensures that the next generation of DNA will not only be the same as the parental generation, but will also contain one strand from the parental generation as shown in the red, therefore making DNA replication semi-conservative. This semi-conservative replication ensures that continuity is preserved during DNA replication. Now moving on to the central dogma. The central dogma of biology is the flow of genetic information within a biological system. The information from DNA is transcribed to make mRNA, which in turn is translated to create protein. First of all, DNA must be replicated. The replication of DNA starts at a site called the origin of replication. An enzyme called helicase, shown here in green, unwinds the two parental strands to allow replication to occur. The sites where the parental strands are being unwound are called replication forks. As the strands are separating, single-strand binding proteins attach to the unpaired DNA strands to stabilize them, shown here in purple. Because of the tight strain put on the DNA as the strands unwind, an enzyme called topoisomerase, shown here in yellow, helps to relieve it by breaking, swiveling, and rejoining DNA strands. Because enzymes that are responsible for synthesizing DNA can only add nucleotides to an existing chain, an RNA primer is established as the starting complementary strand by an enzyme called primase, shown here in red, so that enzymes can attach to the primer and begin replication. For the sake of space, helicase, topoisomerase, and the single-strand binding proteins were removed, but in actual replication, they do remain. The enzyme DNA polymerase III, shown here in yellow, which is responsible for adding nucleotides for the complementary strand during replication, can only elongate the strand in the 5' to 3' direction. Along one template strand, DNA polymerase III can continuously synthesize the complementary strand. This strand is called the leading strand, and it synthesizes the nucleotides in the direction toward the replication fork and is shown here. But because DNA is antiparallel, DNA polymerase III has to synthesize the new complementary strand of the other template strand away from the replication fork. This strand is called the lagging strand. It cannot elongate the strand in a continuous manner and must synthesize DNA in fragments, called Okazaki fragments, which can be seen later on. Multiple primers put by primase, shown here in red, are used to accommodate the discontinuous elongation of the lagging strand. DNA polymerase III, shown here in yellow, then adds on DNA nucleotides, continuing from the primer. The RNA primers are then replaced by DNA polymerase I, shown here in green, with nucleotides. However, DNA polymerase I cannot bridge the final nucleotide of the DNA fragment that replaced the primer to the first nucleotide of the adjacent Okazaki fragment. Therefore, an enzyme called DNA ligase, shown here in blue, comes and joins the sugar phosphate backbones of all the Okazaki fragments into a continuous DNA strand. This concludes DNA replication. Now we move on to transcription. DNA is then transcribed, and there are three stages of transcription: initiation, elongation, and termination. For initiation to begin, transcription factors must bind to the promoter region of the DNA, which starts from the beginning of the green to the end of the yellow. In order for the transcription factors to bind to the promoter region, they must first recognize the green top box. Once the first set of transcription factors bind, then another set of transcription factors bind to the DNA, along with RNA polymerase II. All the transcription factors and RNA polymerase II bound together is called the transcription initiation complex, as shown here. Once the polymerase attaches, the DNA strands unwind to allow the enzyme to begin transcription. In the second stage, called elongation, the enzyme continues the unwinding of the double helix and adds nucleotides to the growing end of the RNA molecule, as shown here in red. As the enzyme continues to synthesize the molecule, the RNA peels away from the template and the double helix reforms. The region being transcribed is called the transcription unit. Then, in the third stage, termination, the RNA polymerase II transcribes a sequence on the DNA called the polyadenylation signal sequence, shown here in blue, which codes for a polyadenylation signal in the pre-mRNA. Then, after adding a few more nucleotides after this signal, the proteins associated with the growing RNA transcript, shown moving in yellow, cut it free from the polymerase, releasing the pre-mRNA. And transcription and translation are referring to the eukaryotic processes. Now we move on to DNA processing. Enzymes in the nucleus modify the pre-mRNA before it is sent to the cytoplasm. During RNA processing, first the 5' end of the RNA molecule receives a 5' guanine cap, shown here in yellow, and then the 3' end receives a poly A tail, shown on this end in yellow. These modifications help the mRNA to be exported out of the nucleus and protect it from the degradation from enzymes and help it attach to ribosomes once it reaches the cytoplasm. Because the mRNA molecule has introns, shown here in purple, which are non-coding segments of nucleic acid, they need to be cut out, and the express regions called exons, shown here in red, must be spliced together to form one continuous strand of functional mRNA with only coding regions. Therefore, RNA splicing takes place, which involves particles called small nuclear ribonucleic proteins, or SNRPs. Those are the purple circles with the red squiggles. These recognize the splice sites at the ends of each intron. Several different SNRPs join together with additional proteins to form a structure called a spliceosome, shown here, which interacts with certain sites along an intron and takes the intron out and joins the two exons. And although not shown here because the book only shows a diagram for a segment of RNA, the poly A tail remains throughout all of this. Now onto protein synthesis and how it's regulated. In alternative splicing, one RNA strand can give rise to two or more different polypeptides, depending on which segments are labeled as introns and which are labeled as exons. So here we have the same mRNA strand with only different exons in red and different introns in purple. Therefore, it will give rise to a different protein than the strand previously shown. The product of the protein synthesis depends on the mechanism that decides what regions of the mRNA are exons and what regions are introns. Now onto translation. Translation can also be divided into three stages: initiation, elongation, and termination. During initiation, the small ribosomal subunit, shown here in purple, and initiator tRNA, shown here in green and purple, which is already bound to the subunit, binds to the 5' cap of the mRNA and moves downstream along the mRNA until it finds the start codon. Each red block here is a codon. In the P site to begin translation. The initiator tRNA should already have the amino acid bound to it, but was not put here because we ended up forgetting about it. The union of the mRNA, initiator tRNA, and small ribosomal subunit, followed by the attachment of a large ribosomal subunit, completes the structure called the translation initiation complex, facilitated by proteins called initiation factors. As said previously, initiator tRNA starts at the P site, and the A site is available for the next tRNA to come in, which is also shown here in green and purple and is moving in. During elongation, amino acids are added one by one to the growing polypeptide chain by tRNAs in a step-by-step process. First, the anticodon of the tRNA recognizes the codon of the mRNA in the A site, facilitated by the hydrolysis of the GDP. After that, the ribosome translocates the tRNA in the A site to the P site, which this requires GDP, while the tRNA in the P site moves to the E site, where it is released, and the mRNA also moves along, bringing the next codon to be translated into the A site. The cycle continues until it reaches the end of the mRNA sequence, and the third stage of translation is initiated, the termination stage. In the termination stage, the stop codon is reached in the A site, and a protein called a release factor, shown here in yellow, binds to the codon. The factor promotes the hydrolysis of the bond between the completed polypeptide and the tRNA in the P site, which releases it from the ribosome, and the remainder of the translation assembly comes apart, facilitated by other protein factors. Now we move on to mutation. Here we have a normal gene that codes for normal hemoglobin, as shown in blood with the normal human blood cell. However, the allele that causes sickle cell disease, which results from a missense mutation, is mutated by a single DNA base pair from CTT to CAT, which results in a complementary strand of GTA, which results to an mRNA strand, shown here in red, with GUA, and mutant hemoglobin has valine instead of glutamic acid as previously shown. The alternation of a single amino acid in a crucial area of a protein leads to an alternation of a protein activity, in this case, part of the hemoglobin. The mutation impairs the protein and prevents it from functioning properly. Because of the mutation, the gene was not expressed properly and led to the alternation in protein shape and in protein function, as shown here in the sickle cell-shaped red blood cell.